Hi, welcome back. It's good to see you for week 2 of our Applied Food Science course. Once again, I am Chris Potts, and I will be your food scientist for today. Today, we're talking about Chapter 15, which is the 4 basic food molecules. Now, there are millions and billions of different kinds of molecules in the world, in the universe. But we're not going to use all of them in cooking, so we're going to focus on 4 which are very applicable. That being water, fats, carbohydrates, and protein. Now, you've probably heard of each of these on their own and have some idea of where you find them, what they do, different properties they might have, but we're going to talk down at the molecular level. What is going on, and how is it that affects what we eat, how we cook? So let's start off with one of the most abundant sources on the surface of the earth. Covers about 2/3 of it, and that is our good friend, water. Dihydrogen monoxide, H2O, whatever you want to call it, it's always still refreshing, and an integral part of cooking, because most food is actually mostly water. Our bodies are about 2/3 water. If you get a piece of raw meat, it's about 75% water by mass, and vegetables and fruit can be up to 95% water. So obviously it's an incredibly important cooking molecule, because not only do we use it to cook things, but it's also an integral part of the things that we're cooking. So let's look at the actual molecule itself. Looks a little something like this. We've got an oxygen, which is negative, and we've got a couple hydrogens down here, which are each positive. Now, those charges are what make water so unique and so integral to life on Earth. And the reason is because with a negative up here and positives down here, this is what we call a polar molecule. That is to say that the charge is not evenly balanced throughout the molecule. What that causes is the top of this to be more negative and the bottom to be more positive. And with that, if there are other waters around, the positive parts of these waters are going to be attracted to the negative parts of these waters. And they are going to form a sort of bond. Not the same type of bond that we talked about last time. These molecules aren't going to be permanently connected. This is what is called an intramolecular bond. That is to say, a bond between 2 molecules. And there's a special name for this type of bond, cleverly named a hydrogen bond. That is to say, when the hydrogen of one molecule is attracted to the negative atom on another molecule. Now, water forms all sorts of hydrogen bonds, and it can form lots and lots of different hydrogen bonds with lots and lots of different waters. And this is what makes water so special, because it is so attracted to itself. It has lots of neat little properties that are unique just to water. For example, have you ever noticed that when you dip your finger in water, the droplet will actually stay on your finger? Why is that? Well, it turns out that all the water molecules are so attracted to each other that it stays up on your finger. It's defying gravity. That's how strong these hydrogen bonds are. And that is incredibly important when it comes to cooking, because it really does 2 things. First off, when we heat up water, we can put a tremendous amount of energy into water with a very small amount of temperature change, which sounds very frustrating, and it can be when you're waiting for a pot to boil, because it can take so long, because it takes so much energy to get that water to a place where it's boiling. However, it can also be incredibly useful, because once that water has that energy, it keeps that energy, it retains that energy very efficiently. So once you have a pot of boiling water, it's got a lot of energy to transfer to the food inside of it. And second, it's not going to cool down very quickly, very easily. That's wonderful. Now, the second wonderful thing about water, and it having these very attractive positive and negative parts, is it is what we call a universal solvent. Now, a solvent is just a fancy science word for saying it can dissolve stuff. So if you put most substances inside water, it will dissolve. And what's great for that is when we're talking, for example, soups, broths, anything that is water-based, we can get a lot of flavor compounds out of the food we're cooking and into the water, because those flavor molecules have positive and negative sides to them that the positive and negative sides of our water are going to grab onto and pull out of the food, making for a lot of extra deliciousness. And that's a concept that we'll come back to in later lessons. But let's keep moving. So we've got our water. Second most important in cooking is probably our fats. Now, fats come to us usually in two forms. We've got our liquid fats, which we call oils, and we've got our solid fats, which we call fat. For example, butter or lard. Now, the technical science term for fats that we cook with is a triglyceride. So you might be like, what is a triglyceride? That sounds ridiculous. Well, to talk about that, let's talk about what fats are at their core. So fats are made of two molecules, carbons and hydrogens, and they're bonded together. Now, before we go any further, you should know something about carbon. Carbon can be a bit of a player. In fact, he is a very specific type of player in that he always likes to be bound to 4 other molecules. He likes to have 4 bonds. So as we go, I'm going to be talking about carbon bonding to other carbons or to oxygens. What you need to know is that it's always bonded to 4 things. And if I don't specify, or somebody doesn't specify what the extra things are, it's usually hydrogen. Just an assumption we're going to make going forward. Now, back to triglycerides. What are they? Well, tri obviously means 3. That is referring to a fatty acid. Now, all a fatty acid is it's a chain of carbons bound together. So let's pretend on this piece of construction paper that each peak and each valley, there's a carbon here. And they're each bonded to each other. Now you'll notice that means each carbon is bonded to 2 other carbons. Guess what? There are hydrogens bonded to each of those carbons, specifically 2 of them, because they each have 2 bonds already. Now, each of these hydrogens is so small and has such a minuscule charge, we're going to ignore it. They basically cancel each other out. So, for our intents and purposes, fats, or lipids, if you will, are non-polar. And that's what makes them very special and kind of the antagonist to what water is. Because of their non-polarity, they don't dissolve in water. Which might sound like, oh no, how are we going to get them to dissolve in things? Well, hold your horses for a second. Because consider, in chemistry, in the world, there's a saying, like dissolves like. Now, water is great at dissolving lots of stuff as long as that stuff is polar or has some charge to it. That means there's still going to be flavors. There's molecules in the food that we're cooking that we can't get out of the food with just water. They are insoluble, meaning not being able to be dissolved. That's where oils or fats can come in. Not only can they pull out a lot of the flavors that are there, but they can enhance a lot of the flavors, because guess what? Humans love the taste of this little guy. So anyway, that's the tri, this fatty acid. That is a single piece of this. Well, glyceride is a word for the glycerol molecule. Now, glycerol is just 3 carbons bonded together. But what that does is it makes a little hat for our fatty acids. So when a glycerol bonds to 3 fatty acids, this is called a triglyceride. Now, in both plants and animals, anytime we refer to a fat that we're going to cook with, it is always a triglyceride. You might be asking, well, aren't there diglycerides or monoglycerides with 2 or 1 fatty acid chains? Absolutely! But they have very different properties. We don't use them for cooking. Don't worry about them. Okay, so let's look at our triglyceride here. All right? What you'll notice is we have, each of these has their little kinks to them. They're bonds, they're carbons at the corners, and they're all bonded to each other. So you might be asking, well, if this is just how it is, how do I have so many different fats? Why are there oils? Why are there so many kinds of oils? There's vegetable oil, canola oil, there's olive oil, there's butter, there's lard, there's margarine. What is the difference? Well, there are two big differences when it comes to fats, and they very much affect how these fats act, both in cooking and on their own. First, how long the fats are. Fats can be anywhere from about 10 to 50 carbons long. So their length plays a huge part in this. And the main reason is the bigger they are, the harder it is to boil them and separate them from each other. The other big difference is this is a secret, we don't just have to have single bonds here. Carbon can do this really cool thing where instead of just single bonding to another carbon, it can double bond. What that means is now that it's got one bond to one carbon and two bonds to another one, it only needs one hydrogen, and that changes the shape of our molecule. Okay? Now when we refer to this, and we talk about how many hydrogens are there, how many double bonds, we have terms for this that you've actually probably heard. If there are no double bonds, if every carbon has two hydrogens, it is what is referred to as a saturated fatty acid. Now what if there's a bond? Then it becomes unsaturated. And why are these special? Well, because when there's a double bond, it changes the shape of our molecule. See how right here in our double bond, we get a little sort of tub, a chair, if you will, in our molecule. This changes the shape, and it changes not only its physical properties, but how our body processes it. In fact, when you think of unsaturated fatty acid, you may have heard the term omega-3 fatty acid. What the heck is that talking about? All that means is it's telling you where the double bond is. Omega means the end, and 3 is how many places from the end. In fact, here I've made a little model of an omega-3, because if we count the kinks, we've got 1, 2, and then our double bond is on the third bond. Neat. Omega-3 fatty acid. Very good for you, especially your brain. Okay? Now, unsaturated fatty acids, there can be one double bond, there can be two, there can be several. We usually refer to one bond as a mono-unsaturated and multiple, more than one, as poly-unsaturated. All right? Now, what is the big difference between these two? Well, usually, plants produce more unsaturated fatty acids. Animals produce more saturated fatty acids. And if you think about it, most plant fats are liquid, like this, olive oil, whereas most animal fats are solid, like butter or lard. Why is that? Well, let's look at their structures real quick. If we have these two saturated fatty acids, they can undergo what is called zippering. Now, the idea behind zippering is that these two fit together so well, this is almost like a zipper, right? When you zip it up. Now, these are so comfortable together, and we can add another one. All of these on top of each other, this is a solid. It's very stable. These aren't going, remember we talked about solids and liquids last lecture. Liquids, everything is moving around each other. These aren't really going to move very much. They're very comfortable in their nice little seats there. Whereas when we talk about unsaturated fatty acids, these don't fit together as well. They can still move around each other very much. And that is why most plant fats are liquid. Now, a point to bring up here. There are not any fats that are 100% saturated. There are not any fats that are 100% unsaturated, at least not naturally produced. Now, this creates a problem for a lot of manufacturers because liquid fat is great and all, but us Americans, we love us some butter. It tastes good, right? You can spread it on whatever. Try spreading olive oil on your toast. Not so great. But the problem is those come from animals. Butter comes from a cow. So it's harder to make. So years ago, people had an idea. What if we could take these unsaturated fatty acids, we could shoot hydrogen at them, we could break this bond, we could flip it around and make ourselves a nice saturated fatty acid. Well, what ended up happening is we got these things called trans fats. And the idea is when you bombard an unsaturated fatty acid with hydrogen, when you shoot it at it, you can get the chain to switch around, to take on this configuration of a saturated fatty acid. However, you still have a double bond. The carbon just flips the other way. Now this is great if you want a more solid fat, like a butter. That's what margarine is. It's artificially made solid fat. Here's the problem. Your body is used to these, used to saturated fatty acids. It can process these. That's great. However, we have a hard time with this kind of double bond. In fact, this is called a trans double bond. That's thus the trans fats. That is why trans fats are so bad for you. Your body can't process them. It can't break them down because it thinks that it's a saturated fatty acid, but actually it's unsaturated. So these things just stick around and they clog up your arteries. Not so good. Anyway, great thing about oils is that they're usually liquid at room temperature, and they have a very high boiling point. Meaning, if we want to cook something at a really high temperature, guess what? Oil is the thing for the job, because it's going to stay a liquid even at those very high temperatures, whereas water is going to boil at 212 degrees Fahrenheit. Actually, here in Arizona, it's about 210 degrees Fahrenheit because we're a little above sea level. That's another show. So, oils, fats even, solid fats like butter, they have a very high boiling point. They're not going to turn into a gas unless you put a lot of heat in there, right? The problem with them is that they will reach what is called their smoke point. Now, while you can put a lot of heat into oils and they'll stay a liquid, the reason for that is because they are so big that even if you crank a lot of heat in there, this thing is huge and it's hard to get it to jump off into the air. Well, the problem is the bonds that make up the oils, not as strong, not as heavy. So what happens is as you heat the oil, it'll start breaking these bonds. And then you get these little tiny pieces. Now these have a much lower boiling point. And so you start getting these floating up. Now a lot of people think when you reach the smoke point of oil that it's actually the oil burning. Not the case. It's actually the oil degrading, becoming rancid. Rancid, not a term for smelly stuff. It just means oils breaking down or fats breaking down. This becomes rancid, the fats break apart, and then you've got these little fat molecules jumping out, up and out. Well, here's the problem. This little guy that gets left behind, not so much good eats. This tastes pretty bad. So, moving on from oils, we've got two last categories. We're going to get through them real quick. First, we have the carbohydrates. What the heck is a carbohydrate? It's just what it sounds like. Carbo, meaning carbon, and hydrate, meaning water. It is a combination between carbon, hydrogen, and oxygen. And what happens when you have a carbohydrate is it forms a little ring like this. Usually, in all food, it's going to be these 6 carbon rings is what we call them, where you have 6 carbons, and they bond together with themselves to form a little ring. Now, what's this little ring all by itself, which is going to have 6 carbons, it's going to have 6 oxygens and 12 hydrogens. If you want to look up the structure, there's a picture of it in your book. This is referred to as glucose, and it is known as the basic building block of all other carbohydrates. Now this, what's really handy, it can bond with other carbohydrates. It can form a bond and stick these together. Now, our body is not used to processing a whole range of carbohydrates. We're most used to processing single carbohydrates and double carbohydrates. This being glucose, when you combine them, depending on whether you put this on top or on the bottom, you can get things like fructose, sucrose, galactose. There's a huge range of them, but the important thing is that you have a monosaccharide here. That is to say, a single sugar. Disaccharides we can deal with. Awesome. However, trisaccharides, quadsaccharides, pensaccharides with 3, 4, and 5 chain sugars, we can't process them. So if you're eating foods that are really high in them, the saccharide will get all the way to your gut, and your bacteria will end up digesting it. Guess what? Makes you gassy. But that's a different topic. So back to these. We've got our carbohydrates, right? And they can bond together. Now, while we can't taste very well anything bigger than about a two-ring system, these things can form huge, long chains together. And that is called a polysaccharide. Now, the most famous polysaccharide that we deal with is this guy right here, flour. And flour, as a lot of you guys know, is mainly useful to us because there's this thing in it called starch. Now, what is starch? All starch is it's a long chain of a bunch of sugars, a bunch of carbohydrates, all in a chain. Now, this can take two forms. We can have our straight form, which is known as amylose. This doesn't show up so much. Much more common is amylopectin. Now, this bad boy is made in plants, and essentially you've got a bunch of carbon rings, a bunch of carbohydrates going up the middle here, and it branches off. The benefit of this is that you can add a lot more onto here than you can here. With this, you can only add one at a time. With this, you can add a whole bunch. So if your plant's really good at making sugar and you need to store it somewhere, starch, specifically amylopectin, is definitely the place you want to do it. Now, starch is great because all of these amylopectins are trapped inside these little starch granules. And when you cook with flour, what happens is these starch granules, they burst open. And you get a bunch of these amylopectins all combining with each other. And it forms this sort of mesh. And it's actually going to, there we go, cause this little mesh. And this is what thickens things like soups or sauces, okay? When you make a roux before you make a sauce, all you're doing is you're heating up those starch granules and you're getting all the amylopectin to burst out and form this little gooey mesh that is not only going to stabilize it and give it a nice thickness, but also trap a lot of those flavors that are trying to escape out of your sauce. Really good stuff, carbohydrates. Now, our final molecule that we're going to talk about is protein. Now, protein is, when you look at your cells in your body, protein is the thing that does all the stuff. Now, that sounds very vague because it's a huge amount of things that proteins do. Proteins do everything. They're what's responsible for the color of your hair, the color of your eyes. They're the things that when you make a fist, guess what? It's a protein walking along a different protein that is causing your muscles to contract. When you get a cut, it's proteins being released from those damaged cells that cause white blood cells to come and fix it. What are they fixing it with? Proteins! Proteins are so useful. They are everywhere. They're one of the universal things. And one of the unfortunate things about humans, we can make a lot of different proteins ourselves, but there are 9 essential amino acids we can't make. So, we need to eat protein from other places. What's a good place? The best place? The chicken egg. Now, specifically, the egg white of the chicken egg. Now, the egg white of the chicken egg has actually been labeled as the gold standard for protein. Why? There are lots of things that have as much protein as an egg white. However, the egg white is in just the perfect shapes, perfect stabilization that when we ingest it, we can absorb it really, really, really well. So the absorption compared to the amino acids and proteins that are in this egg make it the gold standard. Quinoa is a close second. If you haven't tried it, look it up. It's pretty delicious. Now, what is a protein? Now, a protein is a string of what are called amino acids. Now, amino acids are the building blocks of proteins. All right? Now, when you get a bunch of amino acids together, you form a long chain called a polypeptide chain. Why polypeptide? Peptide is just another word we use for amino acid. Now, this may seem pretty useless on its own, but here's the thing. Amino acids, since there's all these different amino acids along this polypeptide chain, each of them have different properties, different things they like to be around. Some like to be around water because they're polar. Some like to be around oils because they're nonpolar. What that's going to cause is this will fold into a specific shape that does a very specific function. Maybe it folds into a little mouth that'll chomp up other proteins. There are lots of different things this can do. Now, when it comes to cooking, what does that mean for us? Well, proteins are one of the best things in cooking because they can do so many different things, and they can change what they do based on how hot they are. Now, what do I mean by that? Well, when you have an protein all wrapped up, there are two ways that this can unravel, or denature is what we call it. First, you can heat this bad boy up. When you heat this guy up, you give him enough energy to overcome any wants or needs he has to, say, be around water or oil. Now this guy's got so much energy, he doesn't care, and so he unravels. The other way we can do it is to add acid. Now, an acid or a base can create such a hostile environment that the outside amino acids basically say, no, I don't want to be around you, and it unravels so that everybody's equally miserable. All right? So what benefit could this possibly have to cooking? Well, there's a few. First off, sometimes we have what are called protein complexes, which may be multiple proteins together. Now, let's say we want to separate these proteins because they each have a really nice taste or texture that they add, but they're all tangled up. If we denature these proteins, we heat it or we change the acid environment. You can even do this physically. When you beat an egg, you are actually denaturing proteins. And it can cause these to come apart. Another example is, let's say there's a part of this protein that you want to interact with another protein, because these two, when they combine, are super delicious. But they won't interact right now. Well, if we denature this protein, we get it off from around its little binding site and we denature this guy. Well, now the parts that we want to interact can. This is the main reason that we ever want to cook and alter proteins, mainly because when they denature, not only did they create unique, new, delicious taste. For example, when you brown meat, you are denaturing the proteins on the outside of that meat, and dear Lord, is that delicious. But also, what happens when these things denature? All of a sudden, you've got these long chains. These, again, form a little mesh. And what's great for us when we're cooking, say, eggs or fish or steak, this mesh will actually trap water inside of it and keep it from escaping. So when you have a perfectly cooked fish, it's so moist and flaky. And the reason is because this has formed this nice little mesh with all these other proteins, and now there's water trapped in there. It makes it delicious. However, if you overcook this, if you keep heating it, basically these proteins will start squishing together all together, and you're kind of reversing the denaturing process, and it's going to squeeze out any water that was in there, and guess what? Dry, gross fish. Ew. So that's proteins, that's carbohydrates, that's water, and that's fat. I hope you learned something, you understand those a little better. In fact, your lab this week is going to be looking at specifically each of these, how they interact with heat, how they change going up and down, and a little bit about our two molecules that can be cooking mediums, specifically oil and water. You're going to start by heating up a little bit of oil, seeing what's going on, a little bit of water, same thing. And you're going to do a little sugar. Now, warning on the sugar. When you heat up sugar, it can be, well, let's just say in the culinary world, we call it culinary napalm. Not so good. Definitely don't want to get it on you. Please be very safe. Because of that, in the lab, there are some precautions I've advised, including when you start, you're going to add about a tablespoon of water, about equal water to sugar ratio. That way, when you heat it up, you'll still be able to see the chemical change going on in the carbohydrate, in the sugar, but it's not quite as volatile. Now, if it sticks to your pan, add a little bit of water, let it sit for a little bit, you'll be fine. Okay? The second part of this, you're actually going to be cooking an egg. Specifically cooking an egg to observe the change in proteins and observe our denaturation, our coagulation when the proteins come together. But you're going to be cooking it two ways. One in water and one in oil. To see how these, as cooking mediums, rather than being in the food, to cook the food, how that affects what is going on in our food. Now with that, thank you for watching. We'll see you next time.